Okay, in this video I want to talk about induced pluripotent cells research. Now, in 2006, a researcher at the Uni of Kyoto, Kyoto's in Japan, discovered a way of returning mature skin cells from a mouse to their pluripotent state, returning them from specialized cells back into the original state, And he called these cells induced pluripotent skin cells. I for induced, small I, capital P for pluripotent, capital S for skin, capital C for cells, and little s, Why induced pluripotent skin cells? Induced means to cause, So he caused them to become pluripotent again, back to the embryo stem cell stage. Why is it useful? Well, they can potentially be used to produce replacement parts of cells or organs damaged by disease. Example is nerve cell replacement, in Parkinson's disease. And you don't have the problem with rejection of replacement tissue because they can be made from the patient's own cells. So stem cells can be taken from, say, fat deposits in your leg and be used to treat disease. In animals, scientists have shown that they can cure. They've shown that they can cure a genetic disease called sickle cell anemia. Sickle cell anemia, if you remember, is that when the cells become sickle in shape, they become like this, and they clog up the arteries, and they cause a deficiency in red blood cells. And unfortunately, people with this disease die at a young age. So how did this guy, this researcher at Kyoto University, actually do this? Well, here's how we did it. So what he did was he got 4 genes, so 4 sections of DNA. So he got 4 genes, and he introduced these 4 genes into a virus's DNA. So he chucked them into a virus's DNA, and then the virus infects the mouse skin cell, and it introduces new genes into the mouse DNA, right? Because the genes have been introduced to that. And so what you get is the DNA. This is the DNA. Think of this ball as the DNA, and it's got you've got the mouse skin here, and you've got the mouse DNA here, right? And then you get induced pluripotent skin cells. So you're going from specialized to not specialized. And the proteins are coded by by these genes. They induce the mature skin cells to become iPSCs, right? So the proteins coded for by these genes they cause the mature skin cells to become induced pluripotent skin cells. You're going from specialized back to the basics. So that's that. Let me now talk about in vitro fertilization in a bit more detail. IVF. In vitro fertilization, IVF for short. I'm sure you've heard of this. It's basically the process of fertilizing the eggs, as I mentioned, outside of the body and placing back the embryo into the woman's uterus or womb. Okay, so fertilizing eggs outside body, then placing embryo back into uterus or the womb. Okay. It can be used. It's used to treat couples who are infertile or having trouble conceiving a child. Okay, so used to help treat couples who are infertile, and God forbid this from happening to anyone. I actually have an uncle who's been using IVF and he's been struggling. It's not a nice thing at all. Trouble conceiving a child. Okay, naturally. Here are the possible consequences of infertility. Okay, so infertility can cause a blockage of the fallopian tubes in the female reproductive system, and it can prevent the gametes, the female sex cells, from meeting. Right, so that's one possibility. Also, a disease that can cause damage to the ovaries. Let's say the woman has ovarian cancer, for example, she can't conceive. If there's abnormal or insufficient sperm from the male. Okay. Also, blocked sperm ducts in the male. Okay. It's not new technology, IVF. It's actually been used in rabbits since the 1940s and 50s, right? And humans, it was first tried in humans in 1965, where the first IVF baby was actually born in England in 1978. Okay, and it turns out about 1% of the Australian population has been conceived using IVF. What are the problems with IVF? Well, it's very expensive, right? And it's sometimes not guaranteed. So that's IVF.